Next, we're going to compare skeletal muscle, cardiac muscle, and smooth muscle. And I want to draw your attention back to table 9.3 in your textbook that has a very detailed comparison of these three different types of muscle tissue. So as you recall, skeletal muscle is associated with the skeletal system. Cardiac muscle is found in the walls of the heart, and smooth muscle is found in many places. We're mainly going to be looking at the smooth muscle found in the hollow body organs, like your digestive tract or like your blood vessels. The cell shape and appearance is very different. Skeletal muscles are very long, multinucleated, with really obvious striations. Cardiac muscle has usually just one nucleus per cell. Uh, and the striations aren't quite as obvious, but they still are striated. Smooth muscle is very different. One nucleus per cell. They're very short. They taper at the ends. That's called fusiform. And you don't see striations. Likewise, there's a little difference in the connective tissue components. Skeletal muscle, I emphasize the endomysium, perimysium, epimysium, all eventually forming a portion of the tendon. In cardiac and smooth muscle, it's mainly endomysium, holding the cells together. Skeletal muscle and cardiac muscle are both striated and they both have myofibrils. Cardiac muscle, you don't see the striation as obvious, and that's because the myofibrils aren't perfectly uniform. And in smooth muscle, you do not see striations. Now, you still have thin and thick filaments, you still have actin and myosin, but there's a different type of connection. All right. Another major difference between skeletal, cardiac, and smooth muscle is whether or not they have the T-tubules. Both skeletal muscle and cardiac muscle have T-tubules, and they have that relationship between T-tubules and the sarcoplasmic reticulum, with the sarcoplasmic reticulum storing the calcium. But smooth muscle does not have T-tubules, does not have the extension of the cell membrane down inside the cell, but they do have some small infoldings called caveolae. So we're really going to spend most of our time looking at smooth muscle because smooth muscle is so very different than both skeletal and cardiac. Cardiac muscle we will cover in more detail in A&P2. When we get to the cardiovascular system, we have an entire chapter just about the heart. So smooth muscle is mainly found in the walls of most of the hollow organs except the heart. And usually when we see it, it's in two layers that are at angles to each other. One that runs along the length of the organ called longitudinal muscle, and one that encircles the organ called circular muscle. So here's a good example of the smooth muscle in the walls of these hollow organs. So here you can see the hollow organ. So this is a section of the digestive tract. All right. The interior is made up of circular muscle, and so it's running around the area of the digestive tract. Longitudinal muscle runs along the digestive tract, so it's running in this direction. Okay. Microscopically, smooth muscle is very different. It's spindle-shaped cells, so they taper at the two ends. They only have one nucleus and no striations. They don't have all those layers of connective tissue. You just have endomysium surrounding each cell and holding all of the cells together. There's much less sarcoplasmic reticulum, SR, than in skeletal muscle or even than in cardiac muscle, which means that very little calcium is stored inside. There's also no T-tubules. Now, T-tubules were to connect the action potentials with the sarcoplasmic reticulum. Well, since you don't have much sarcoplasmic reticulum, you don't really have T-tubules. But you do have these little infoldings called caveolae, and this is going to allow the cell to pull in calcium from the extracellular fluid. So you don't see the distinct sarcomeres, no striations. You don't have the myofibrils, and you don't have the T-tubules in the smooth muscle. Now, you still have the myofilaments. You still have thin and thick filaments, but they are arranged in a spiral around the smooth muscle. And when smooth muscle contracts, it twists up kind of like a corkscrew. Now, you still have to have the sliding filament mechanism, but now instead of having Z discs at the end of sarcomeres, you have these dense clusters of protein that anchor the overlapping thin filaments to the sarcolemma. So it sort of acts as both the Z disc and the dystrophin molecule. But when you have contraction, you are going to bring the dense bodies closer together in smooth muscle. So here's our little view of a relaxed smooth muscle. You can see the caveolae, the little in pockets at the surface, allowing you to bring in lots of calcium. You also see the gap junctions. Now, gap junctions will be a connection between this smooth muscle cell and a neighboring smooth muscle cell. You also see the filaments connecting together the dense bodies, and when you have contraction, we're going to see these dense bodies coming closer together. So here's a similar cell in a state of contraction. So you can see those dense bodies are being drawn closer together by the sliding filament mechanism. So contraction smooth muscle is usually very slow. Cells could either be energized by the nervous system directly to cause them to contract, but many smooth muscle cells are connected together by gap junctions. So this will transmit an action potential from one smooth muscle cell to the next smooth muscle cell. And some smooth muscle cells are self-excitatory, similar to cardiac muscle. So certain sheets or large groups of muscles may contract and relax in a rhythmic pace. Right. Now, just like in cardiac muscle, how fast this contraction occurs and how strong the contraction occurs can be modified by the nervous system. But we still have in smooth muscle, even though there aren't striations, we still have the sliding filament mechanism. You're still going to have myosin heads that are going to reach out and grab onto actin and push actin towards a center line. Right? And just like in skeletal muscle and cardiac muscle, the trigger is going to be intracellular calcium levels. In this case, you're going to get a little bit of calcium coming from sarcoplasmic reticulum, so there still is sarcoplasmic reticulum, but the vast majority of the calcium is going to come from outside of the cell, from the extracellular fluid. In skeletal muscle, almost 100% of the calcium comes from the sarcoplasmic reticulum, and just like in skeletal muscle, ATP is the energizing process, but we're going to see a little difference in how the ATP is used. All right, so what does calcium physically do? This is where it's very different than in skeletal muscle. In skeletal muscle, calcium attached to troponin and tropomyosin and moved the tropomyosin away from myosin binding sites on the actin. And in skeletal muscle, that was how the trigger occurred. But here in smooth muscle, calcium is going to bind to another protein called calmodulin. Let's break apart that word. Cal for calcium, modu because it modulates or controls contraction, and IN means it's a protein. So calcium binds to calmodulin, activates it, and that activates a myosin kinase. All right, myosin kinase, a kinase, will add a phosphate to a protein. And in this case, the protein that gets the phosphate will be myosin. So the myosin kinase will phosphorylate and activate myosin, and that's how the myosin head gets activated. The myosin head isn't really its own myosin ATPase. You have a different myosin ATPase, myosin light chain kinase. But just like in skeletal muscle, when the myosin head is activated, cross bridges will form with the actin and you get contraction. And then when intracellular calcium levels begin to drop, you get relaxation. So let's look at this series of events, and I know this may be a little bit hard to see on the video, but I want you to notice that this is figure, let me write it out for you, figure 9.28 from your textbook. So you should take a look at that figure in your textbook and see this sequence of events. So mainly we have calcium. So up here we have our calcium coming in from the outside. So it comes in from the outside. Very little will come in from the sarcoplasmic reticulum. This calcium is going to bind to calmodulin. So we go from having an inactive calmodulin to activating calmodulin. That will activate the myosin light chain kinase, so it becomes activated, and that will allow the myosin head to move back to a Cochteau ready position. But then you still have your sliding filament mechanism, so we still have our thin filament attaching to our thick filament for contraction to occur. So contraction in smooth muscle is also really slow. You have many steps, many more steps than we saw in skeletal muscle. So it's slow to contract, slow to relax, but that's really good because you can maintain a partial state of contraction for a long period of time without using up a lot of ATP energy. All right. Relaxation requires that the calcium levels go down, so calcium detaches from calmodulin, and then you actively pump the calcium out of the cell, out into the extracellular fluid, and the myosin ATPase activity goes down. A couple of special features of smooth muscle contraction. Smooth muscle contraction really responds to stresses or stretches in the muscle. So when you stretch out the muscle, it's going to be able to contract as a result. So it allows organs like the stomach and bladder, you can stretch it out and store large amounts of material and maintain some pressure on it. And it has the ability to maintain contraction over a really wide range. So there's a much larger length tension relationship than we saw in skeletal muscle. You can change the length of smooth muscle and still have really effective tension produced. And then one last special characteristic of smooth muscle that I want to point out is that it is one of the few types of muscle cells that can actively increase the numbers. So you can divide and increase the number. You can increase the length of the digestive tract. You can increase the length of blood vessels. And you can also increase another really muscular organ known as the uterus. The uterus is mainly made out of smooth muscle. It's a very muscular organ. So estrogen will change the shape of the uterus at puberty, and obviously the size of the uterus is going to become much larger during pregnancy. So you're going to increase the amount of smooth muscle in the uterus. So that's known as hyperplasia. Hyperplasia, just to make sure we get two, because I've used a couple of different terms throughout this entire series of lectures. Hyperplasia is specifically an increase in the number of cells. So organs could get larger because of hyperplasia, increasing the number of cells, as opposed to hypertrophy. So we talked a little bit about hypertrophy when you do weightlifting exercise with muscle cells. Hypertrophy will increase the size of individual cells. So once again, though, you could increase the size of an organ by hypertrophy, increasing the size of the cells. So the uterus, for example, gets larger due to hyperplasia. Your skeletal muscle as an organ will get larger due to hypertrophy, increasing the size of individual cells. So that's going to do it for Chapter 9, the muscle tissue chapter.